Hello guys, how are you? I'm your teacher Umar. I hope you are fine. In this video, we'll talk about DNA, chromosomes, and genes, and we will study in different lecture videos. So this is the first part. I hope you will learn a lot from this lecture series. If you haven't subscribed to my channel, please subscribe it, like it, and do tell me in comment how you will like my video. So starting today, DNA, chromosomes, and gene. The life depends upon ability of cell to store, retrieve, and translate the genetic instruction required to make and maintain living organisms. So it is the duty of cell not only to store, retrieve, and translate genetic instruction, and some cell has the ability to pass on this genetic instruction to the next cell. The hereditary information that how traits are transmitted from one generation to another is passed from cell to its daughter cell at this stage of cell division, and from one generation of an organism to the next through the organism reproductive cell. So there are two types of cells that undergo division. There are somatic cells and germline cells. The division of somatic cells will refer to as individual cell, but the division cell division in germline cells is ensures that how genetic information is passed on from one generation to another. The instructions are stored within every living cell as its genes, the information containing elements that determine the characteristics of species as a whole and of the individual within. So genes are one of the most important things that are involved in passing of genetic information. So we can say that if the hair color of an individual is passed to its progeny, the genes are solely responsible of doing that. When genetics emerged as science at the beginning of 20th century, scientists became intrigued by the chemical structure of genes. You know, late 19th and early 20th centuries is the revolution in the genetics, molecular biology, genetic engineering, biochemistry. A lot of things happening. The information in gene is copied and transmitted from cell to the daughter cells millions of times during the life of multicellular organisms. This is pretty obvious. The genes that are copied transmitted from cell to daughter cell millions of times. You know, the somatic cells, there are millions of cells are dividing daily into daughter cells. For example, your hair are solely responsible when you cut the hair. You got a haircut, you will get after a few times you will get your hair back. Same goes to nails, RBCs. The lifespan of red blood cells is 120 days, and there are hundreds of examples that this illusion is carrying on enormously. What form of molecule could be capable of such accurate and almost unlimited application, and also to exert? Scientists are trying to know what kind of instruction does the genetic information contain, and how can enormous amount of information required for development and maintenance of organisms fit within tiny space of cells. So this is one of the important questions scientists were trying to figure. The answer at several these questions began to emerge in 1940 when the researchers discovered from studies in a simple thing that genetic information consists largely instructions for making protein. Discovery of protein is one of the important contributions in early genetics and molecular biology. Proteins, they are phenomenally versatile macromolecules. They perform most cell functions. Enzymes are proteins, antibodies are the protein. The proteins are building blocks, cell structure from the enzyme that get live most of the cell by chemical reaction. They regulate gene expression, enable cells to communicate. The properties and function of cell and organism are determined to great extent by protein they are able to make. This is the observation of painstaking the cells and embryo in the late 19th century had led to the recognition that the hereditary information is carried on chromosomes, the strand-like structure in the nucleus of eukaryotic cells that become visible by lack of copy as cell begin to divide. And we can see that the chromosomes are visible only in dividing phase. This is the diagrammatic explanation of dividing and non-dividing cells, and the two adjacent plant cells. They are photographed through light microscope. The DNA is stained with the fluorescent dye DAPI that bind to it. DNA present in chromosomes which become visible as distinct structure in light microscope when they become compact sausage-shaped structure in preparation for cell division, shown in the left. You can see cell on the right which is not dividing contain a radical chromosome, but they cannot be clearly distinguished at the phase in the cell life cycle because they are more extended conformation. And the B is the schematic diagram and of the outline that two cells along with the chromosome. This is the Peter chart. Thank you. When biochemical analysis become possible, a chromosome or found which consists of deoxyribonucleic acid protein with both peak present roughly in the same amount. For many decades, the DNA was thought to be mere a structural element. However, the crucial advance made in 1940 was identification of DNA as a likely carrier of the genetic information from gene, chromosome, and now DNA is considered to be the responsible for carrying of genetic information from one generation to another. The breakthrough in understanding of cell came from studying inheritance in bacteria. This is one of the most important experiments. These experiments carried out in 1920, the first one and in 1940 B. They showed the adding the purified DNA to the bacterium changed the bacterium properties and that this change was faithfully passed on to the subsequent generation. Too closely related strain of the bacterium Streptococcus pneumoniae differ from each other in both their appearance under the microscope and their pathogenicity. One strain appears smooth and other and cause death, while other infecting to mice and they appear to without and is non-lethal. An initial experiment shows that some substance present in that strain can change or transform the R strain into the F strain, and that this change is inherited by subsequent generation of bacteria. The B experiment, in which R strain has been incubated with various classes of biological molecules purified from the S strain, identify the active substance as DNA. This is S strain random mutation causes R strain rough non-pathogenic mutant bacteria. Live R strain cell grown in the presence of either heat kills S strain or cell-free extract of S strain. Then transformation was done. Some R strain cells are transformed S strain cells whose daughter or pathogen journey can cause pneumonia. Conclusion: the molecules that carry heritable information are present in S strain. Now this fractionation of cell-free extract into the class of purified molecule, RNA, protein, DNA, lipid, carbohydrate. Only the DNA contains S strain and all other macromolecules have R strain. The conclusion: the molecule that carries heritable information is the DNA. Okay. Later, when biochemical analysis became possible, chromosomes were found to consist of deoxyribonucleic acid protein with both peak present roughly in same amount. Okay. But still in 1950 began both how protein could be specified by instruction in the DNA and how this information might be copied for transformation from cell to cell seemed completely mysterious. To puzzle was suddenly solved in 1953 when Watson and Crick they revised the structure of DNA. They were then given the Nobel Prize for revising the structure of presenting the model of DNA structure. The determination of double helical structure of DNA immediately solved the problem of how information in this molecule might be copied or replicated. It also provided first clue as how a molecule of DNA might use the sequence of its subunit to encode the instructions for making protein. Today, the fact that DNA is genetic material is so fundamental to biological thought that it is difficult to appreciate the enormous intellectual gap that was filled by this breakthrough discovery. Despite its chemical simplicity, the structural and chemical properties of DNA make it ideal to utilize raw material of genes. We then consider how the many proteins in chromosome arrange and package this DNA. This has done in orderly fashion so that chromosomes are replicated approximately correctly between the two daughter cells at each cell division, and also allow access to chromosomal DNA both for enzymes that repair DNA damage and for the specialized protein that direct the expression of many genes. Okay. In the past 2 decades, there has been revolution in our ability to determine the exact area of subunit in DNA molecule. As a result, now know the sequence of 3.2 billion nucleotide pairs that provide information for producing human adults from a fertilized egg, as well as having the DNA sequence for thousands of other organisms. The detailed analysis of these sequences are providing exciting insight into the process of evolution, and it is with the subject that the chapter ends. Okay, the genetic mechanism, ways in which cell make, replicate, express genetic information carried in DNA, how DNA sequence can be arranged, genetic recombination, gene expression. We will discuss all in our upcoming lecture. The structure and function of DNA. I think this is all from today's lecture. We will study in next video the structure and function of DNA. If you learned anything you like this video, please subscribe to my channel and share it with your fellows. We will continue our series of DNA, chromosome and genome. Thanks for watching. Take care and like our video.